So perfect. Can you hear me well? Okay. I'm Axel Fehrenbach. I'm from the University of Tübingen. I'm a PhD student there, and today I'm going to talk about CRISPR array evolution and what we do to track this evolution, or what tool we develop to track this evolution, and which we are hopefully soon publishing. First, what is CRISPR array? What is a CRISPR array? I mean, not everybody is maybe aware of that. CRISPR arrays are found in the genome of bacteria and archaea. So you have this CRISPR array, which is just repeats with interspersed sequences, which are called spacers. And they are actually of foreign origin, not from the bacteria. And they're supported by CRISPR-associated genes, which are actually like the tools that people are using to do gene modification. So, and what is the CRISPR array responsible for? So the CRISPR arrays, or CRISPR system in general, if a virus invades like the cell, then it inserts its DNA, and Cas1 and Cas2 can take up like small snippets of this DNA, which are then these spacers, which are then inserted into this CRISPR array. These can then be replicated by the bacteria and incorporated into some other proteins, which can then go around the cell, find matches, and cut them and destroy them through that process. So in a sense, it's an adaptive defense system, and the CRISPR array is actually the memory of the system. So how does the system evolve? As I already mentioned, we insert new spaces into this array. And what is really remarkable is that these spaces are always inserted at the leader end, like at one end of the array, which actually means that we have a real timeline of spacer acquisition, and thus of phage infections or phage pressures. And if you're inserting stuff, you have to delete stuff, otherwise you won't remain efficient over long times. So there are deletion events, and this happens along all positions of the array, but the deletion process itself is not really well understood. There are some ideas how this could work, or some of these processes. For example, during replication, repeats can stick together and form like a loop in between, and in this loop there can be the spacer, which means that if this is replicated, it's just missing one of these spacers, which is like one explanation, but there are multiple more of what you can think of in that case. So in the end, you just miss it and it's gone. So how do we think about CRISPR arrays, what we are looking at? So first, of course, you have to extract the CRISPR arrays from the DNA. So you have some amount of strains which are closely related and have some overlap in spacers. You use some amazing tools, so it's non-trivial to extract these CRISPR arrays, but there are tools who do it. Afterwards, we simplify the problem a lot to make it more feasible to work with that. So instead of looking at repeats and leaders, we remove them. We're only looking at the DNA sequence first, and then each distinct DNA sequence, each distinct spacer gets a number, a natural number. And that's what we end up with. So that's the data we're talking about. And in the second step, we align these numbers just with math, for example. And you end up with this spacer alignment, which you see on the left, which is just a presence-absence pattern. And each column corresponds to one specific spacer. What important thing to note is that there exist duplicates of spacers in this alignment. There exist rearrangement events and stuff, which are problematic for our method, so we need to deal with them. It's actually the reason why we need to do a spacer alignment, but I'm not going to go deep into what we do with them. They're treated somewhat differently. And the most important fact is that we relabel them at this point in time, so we give them unique names in that case. So I talked about this polarized insertion, and this is really useful because if you look at each of these arrays in the spacer alignment, you see that on the left is the leader end. So you always have for each of these rows, for each of these arrays, you know in which sequence they happen. So you know 15, the orange 15 on the left is the youngest spacer and 1 on the right, for example, in green is the oldest spacer of the first strain here. And then you can also combine all of this information into a graph where you have like the whole information across all of these strains within your data set. Important notice that it's a partial order, so you don't always have relationship between every spacer. For example, if you think about the 15 on the left, it has a relationship to 4, 3, 2, and 1 because we know it's in front, so it's younger, but we don't know the relationship to all of these other spacers which are shown in the alignment, right? Because there's just no connection. So how do we model CRISPR array evolution? So what can we do? So we start with a tree, which we, in most of our work for now, did with the core genome, just a basic pipeline. And so we start with a tree and we start with the spatial alignment. And we develop, or what we didn't develop, that's well known. We use a very simple model, the independent deletion model with unordered gains, which is just a normal time reversible model or a substitution model with 2 states. If you look at the bottom, can you actually see that? Yes. You have your array, and this evolves over time in a continuous Markov process. And you can gain not present spacers with some rate theta, and you can lose every position that is occupied with some rate rho. So very, very simple model for a start. Now we have all of the setup. We have a model, we have some data we can work with, we have a tree, and we want to reconstruct the ancestral states of these arrays along this tree, which is done by a spacer placer. So in the end, we want to end up with something like this, where we can see on the branches in these green circles what spaces were inserted at which time, and in these squares, red squares, which spaces were deleted at which time. So we start with a very simple reconstruction, with the guide reconstruction, with this independent deletion model, because every site is independent, so every column of our space alignment we can treat independent, so it's very easy to compute the joint maximum likelihood reconstruction. There are very well-known methods to do this very quickly. What is very important to notice that we choose the insertion rate much lower than the deletion rate. This is because we want the model to describe the evolution with few gain events and a lot of deletion events or more deletion events in comparison. This is generally, we do this because we generally expect that the spacer array seldomly inserts the same spacer, again, exactly the same spacer multiple times. Because if you think about the genome of a virus, which is some kilo base pairs big, and you have these small snippets, which are around like 20 to 50 base pairs, it's just very unlikely that you hit the same exact sequence multiple times. And as we see, these arrays are actually quite short. They're in length like around 15 spacers on average. Yeah, perfect. So, sorry. So we can already get a reconstruction. So you can see there is some events were placed, but there's a big problem with this reconstruction. If you see, for example, and maybe some people in the back have to believe me, on the top branch, there's an insertion of 15 and 1. And 15 is fine, that can be there, why not? But 1 is actually the oldest spacer, which we already know from this partial spacer insertion order, so it can't be there. So we need to do something about this. So what we do is we detect these contradictions to the pseu, and if we find none, then we're finished, we have a reconstruction. Otherwise, we put up the insertion, or we move the insertion, the contradiction one step above to the parent node by just saying at the parent node, the spacer has to be present. It's important that we are not saying that the spacer insertion event has to happen at this place, but we say that the spacer has to be present there. And then we run again the reconstruction with the simple reconstruction model and with these fixed sites, and we do that until we don't find any contradictions, which in the worst case would be if everything is moved to the root, if something is weird in your data set. So in the end, you start with this guide reconstruction, which has very few deletion events, and you get to something which seems a lot more natural, which is then this refined reconstruction. So what can you do with this reconstruction? There are a lot of different things you can think about, or like the most important thing for us from population genetics or from evolution is, okay, maybe we can study this evolution, maybe we can define some models, look at these ancestry constructions, put them into our model and see what comes out in different likelihoods and estimated parameters. We can look at selection effects. For example, are there some specific spaces that are deleted more often or less often? Are there specific positions in the array which are deleted less often or more often? You can track the microbiome if you have the spacer-phage correspondence, which is not that easy, but there are people who do that. And there are some things like you could think about doing phylogenetics with CRISPR arrays, but I'll come to that again. And in the end, there's a point which I'm going to talk about, which is actually somewhat surprising, was somewhat surprising for us. So let's start with like what we did in space evolution to understand space evolution better. So what we asked ourselves, can't spacers be deleted in blocks? If you think about this very simple deletion model, why shouldn't there be like multiple spacers within this loop? I mean, there's no like real contradiction in that sense. And if you look at reconstruction already a bit earlier, you see these very long stretches of deletions. They don't seem to fit like an independent deletion model where you just lose spaces individually. So what we did, we developed a model. We used a new model, which is the block deletion model. So we're now not thinking about insertions. We're only thinking about deletions. And like the only difference in quotes is that spacers deletions can still happen with some rate at each position in the array, at each occupied position in the array. But now the length of this is geometrically distributed, which essentially means if a deletion happens, you delete one spacer, then you toss a coin. If you hit, you delete another spacer. If you miss, you stop the deletion and you have your deletion length. And this is all described by the parameter alpha, which is like the mean length of this deletion. The nice fact of this is that this is monotonically decreasing in likelihood for length. So the length of 1 is still the most likely event to happen, and length 2 is then less likely, length 3 is less likely, and so on. And the other thing is that if alpha is equal to 1, we're just back in the independent deletion model. So what can we do? We can extract the events we can run our reconstruction algorithm. We can extract all these deletion events and the keep events. We can put them into a likelihood ratio test and now compare between the independent deletion model and the block deletion model. And what's nice as well is that we actually get some parameter estimates for these deletion rates on the side because we're doing maximum likelihood estimates anyway. So now to the results of this. So in short, block deletion model seems to fit or fits better than the independent deletion model, which you see it here on the top left of this thing, which is actually separated by a CRISPR type. Okay, you can't see my mouse anymore. Anyway, so in most cases, the independent deletion model is rejected, which means that spaces are more likely lost in blocks. Are they lost in the block deletion model? We don't know, but at least in some kind of block deletion model, not in the independent deletion model. The other thing I want to point you to is the last point where we have the per spacer side deletion rate, which is roughly rho times alpha, which is around 400. And since we're using trees based on the core genome, which are in the per site mutation rate scale, we actually know that this per site spacer site deletion rate is 400 times higher than the per site mutation rate in this mean or median in this case. Which means that it's just evolving much faster than general normal mutation evolution. Which gives you an idea. Maybe we can do actually high resolution estimation of phylogenetic trees based on CRISPR arrays. So here's an example from a paper from Tomita. They sequenced, or they got sequenced strains from different hospitals, and they did some MLST tree, which you see on the left, and like the CC4 group is the interesting one. And as you can see, there's not much resolution. You don't see anything at all. You can't really separate them. If you look at the CRISPR arrays, which are on the right, you actually see there is some difference. There's not like amazing amount of difference, but there is some difference which you can use to reconstruct a tree based on a very simple model that we used with UPGMA and the block deletion model. Lastly, I want to come to a very nice thing we found by accident. So assume we have like our spacer, we have some spacer arrays that we detected, but we actually don't know which transcription direction now this CRISPR system has, or these CRISPR arrays have. This happens quite a lot because this prediction of the orientation is actually not that easy. But what we can do, or how we can find the leader end of the array, is by just applying our model twice. So we start with the forward direction, so the one we got it in, run the model, extract the events, compute the likelihood under the block deletion model. And the second part is we just reverse the alignment, do the model again, and compare these likelihoods against each other. And then you have to estimate like some threshold that you can use, but then you can make a decision, okay, if it's really unlikely, if you look on the right here, there are a lot of insertions at the root, which doesn't seem really like a good evolutionary model. It should gain spaces in between. And then you can make a decision on that base. So as a conclusion, we have a nice reconstruction tool which works with very simple assumptions, which are like very well based on the biology that we observe. And we can do this, or we hopefully do a lot more applications in very different areas of research. So lastly, I'd like to thank the funding agencies and my supervisor, Franz Baumdicker in particular, and Alexander Mitrofanov, who did a lot of the data extraction for us and was a lot of help during the project. Thank you very much. I'm happy to take questions.
Questions?
Thank you very much. So when I think about the order of the spaces you're observing here, I'm thinking about sort of a health record of the bacteria. And that gives, in my understanding, that should give us some idea of what type of phages do these bacteria encounter more frequently.
Exactly.
Such that they keep the spaces, and which ones are rather rare events they encounter once or twice in their lifetime or whatever, and so they discard the spaces. Is that right?
That's in principle. I mean, you have to also that, like, if you don't have the spacer that is important, then you get killed. So you'll have the spaces that are really important and will be in the array at some point and might be in the back of the array. I mean, there's some people thinking about, okay, where in the array are they positioned? So in that sense, it's just like that. So it's kind of a health record, a record of what happened around the cell, which invasions were problematic, which were not. At least in the end of the array, in the start of the array, I mean, there's some evolution, neutral evolution, right? Maybe.
So the thing I was curious about is how many arrays are in a typical bacterium, and whether there's illegitimate recombination among the arrays, because I could see that essentially if you have two arrays that are different and they recombine, wouldn't that sort of screw up the model by scrambling them?
Yes. So in this experiment we did, where we estimated the rate, we actually excluded arrays where multiple arrays were in the same strain. So if there was a strain which had multiple arrays and they were in the same group, we excluded these arrays because that's problematic. And it's also problematic because we can't estimate a distance between them in our tree, right? In general, there are a lot of arrays in different bacteria. Bacteria can carry multiple arrays. They can carry multiple arrays of the same type with the same Cas genes, but they can also carry a different Cas with different arrays with different Cas genes. I think there's going to be a lot more diversity in archaea, for example, because for them it's a lot less dangerous to have CRISPR arrays. And yeah, so there is within, and there's also, I mean, it's it's an interesting problem. So actually, something we want to look into: what happens if you have two arrays which are in the same bacterium? How do they evolve? Do they evolve differently? Are they combined somehow? Do they specialize in some way? That's an interesting question we want to look into in the future.